Hello, and welcome to the Current Science and Technology podcast from the Museum of Science in Boston. I'm Brenda Muniappan, your host. Every week, we bring you interviews with guest researchers and our museum staff covering science and technology in depth. My guest today is Dr. Sangeeta Bhatia, Professor of Health Sciences and Technology and Electrical Engineering and Computer Science at MIT. Dr. Bhatia recently published a paper about a novel way to use nanoparticles to treat tumors and is here to discuss that research. Hi, Sangeeta.
Hi, Brenda.
Previously in this podcast series, we have discussed a few ways of attacking cancer directly with nanoparticles, these tiny materials that are no larger than hundreds of nanometers, for example. But what's so special about your team's approach to treating tumors with nanoparticles?
We were really interested in the idea that chemotherapy is not very efficiently delivered to cancer at the moment. So of any given injection, only about 1% arrives at the tumor, which means 99% goes elsewhere to cause side effects. So we were interested in sort of like increasing that 1% number. And a lot of people in nanotechnology have been thinking about that idea through what we call targeting. And the idea is that you would take a nanoparticle and then you would load it up with lots and lots of drugs, and then you would decorate them in some way with like a molecular zip code, and that would allow the chemotherapy to kind of home in on the tumor.
And leave the normal cells from being killed by the chemo, hopefully.
So that's the idea, and there's a lot of activity and some nice successes going on in that area, and people are starting clinical trials. But if we look at the numbers, it's actually still not that much better than 1%. So we wanted to think about how to improve that number further. And it occurred to us, myself and my graduate student, Jeff von Moltzon, that maybe we should think about the fact that in any given injection, there are about 1 trillion nanoparticles, not just a few nanoparticles. And that might mean that you could have different populations of nanoparticles, different flavors of nanoparticles.
You can mix it up because you've got so many going in there.
Exactly. So instead of having them all be exactly the same, you could have different kinds and they could be specialized. And if you could sort of figure out how to make them cooperate, then maybe together they could enhance their deposition, increase their accumulation at the site of the tumor. So that idea of having sort of more than one kind of nanoparticle in an injection and having them cooperate to target the tumor is what we think is new about this approach.
Right, that sounds great. And in your paper, you had this nice combination of not only using a variety of nanoparticles, you also used a system that already exists in the body, so a natural process.
Yeah, so we were really inspired by actually the way that your own body is able to identify a wounded tissue. And a really good example of that is when you have an infection. If you have an infected tissue, there are specialized cells in that tissue that give off chemicals, and they tell the other cells in the circulation that there's an infection here. And then those specialized cells, the ones that can fight the infection, can then go and find the infection and mop it up. And so together, these two different particles communicate over pretty long distances in the body in a human living host to sort of perform that function. And so we were kind of inspired by that idea. And so if you think about like how we actually make two nanoparticles communicate, what does that really mean in the body? It really comes down to chemistry. And chemistry is not a signal that can go very far. So we needed to leverage some of the chemical reactions in the living host to allow one nanoparticle to communicate with another nanoparticle.
But you didn't actually use the immune system in this particular case. You used a different system. Tell me a bit about that system and why this one was selected.
Sure. So when we started thinking about what cascades there are in your body, the criteria that we were interested in exploiting was one of amplification. So we wanted to think about chain reactions where the end product has many more events than the initial one. And so there are lots of examples of this. So the immune system is one. There's something called the complement system, which is another way to fight infection in your bloodstream. And then the one that we chose is the coagulation cascade, or the clotting system. And this is a whole network of enzymes that sits in your blood and is normally not active, and they just hang out waiting to be activated by some signal that there's an injury and they need to needy to start getting activated. And once they start getting activated, there's this huge amplification process whereby they activate one after another after another. And at the end of it, you get this huge deposition of clot at the site of injury, which is like a nice cross-linked network of proteins. So you start with a little signal and you end up with a network of millions of proteins. And so that was the cascade that we tried to leverage.
So you sent in nanoparticles to try to initiate a blood clot, is that right?
That's right, and we specifically wanted only to do that in the tumor, because if you initiated a clot elsewhere, then that would actually be dangerous.
That would be very bad, right.
So one of the things we had to do was design nanoparticles that would very specifically activate the clot and not activate it anywhere else in the body. So in order to do that, we used a nanoparticle that could be activated by some kind of external signal, something that the physician would apply. And in particular, the trigger that we used was light. And we wanted to use a wavelength of light that we knew would penetrate as far as possible in the human body. So we chose a long wavelength of light, around 800 nanometers, which is called near-infrared light. And what's nice about this wavelength of light is that it's in what people call the optical window, which means that it won't be absorbed by things like hemoglobin in your blood, and it won't be scattered by other tissues, and it can penetrate pretty far.
So this is kind of the perfect wavelength. It doesn't bother other proteins. It will get into the tissue, but it won't actually damage our tissue. Is that right?
Exactly. Right. It's an innocuous wavelength of light, and you can sort of go home and do the experiment with your remote control and stick it up against your finger, and you can see that, in fact, red wavelengths do penetrate quite far through your body.
So you were able to trigger the nanoparticles that you injected by using near-infrared light, and I assume that began the coagulation process.
Right. So it turns out that if you heat tissues up a little bit, you will get a little bit of injury, and the injury to blood vessels will trigger the clotting cascade. If you heat tissues up even more, then you could actually cook the tumor. Right? So in this case, we're not doing that. We're not cooking the tumor. We just wanted to trigger a little bit of clotting. And what we needed to do was to use a material that would convert the light energy to heat energy. Because again, if you go back to the near-infrared light, the remote control example, that's not actually heating anything. So you need something that acts like an antenna for that near-infrared light to convert the light into heat. And so we used a class of materials known as plasmonic nanomaterials. And they have electrons on their surface that are really mobile, and they resonate with light. And the light wavelength that they're sensitive to depends on their shape. So if you have a spherical gold particle, it would resonate with light or sort of green. And what we used were materials that were elongated. We call them nanorods. And they're about 50 nanometers long and about 10 nanometers wide. And because of that shape, the resonant frequency shifts. And they are now sensitive to near-infrared excitation, which means that only if you shine near-infrared light on them will they heat up. So if you walked around, put green light on them or blue light on or any other light on them, and they wouldn't heat up. Only where you shine a near-infrared laser, which can penetrate through tissue, would they heat up and start the clotting cascade.
So this is important because they're somewhat more safe in this regard, being so specific to the light that will heat them up and actually cause them to start damaging tissue.
Exactly. So we wanted a trigger that was specific so that it would be safe, but also sensitive to the wavelengths that could penetrate through the tissue. So if we had used a green particle, it might have been sensitive, but we couldn't get there with the light that we wanted to shine.
So in this case, you could have stopped right there. You have a nanoparticle, a gold nanorod, that is sensitive to near-infrared light, and you could use the light to excite the particle and have it heat up the surrounding tissue, and that might be one direct way of killing a tumor, but you were looking to amplify that process through the clotting cascade. So there were more steps involved.
Right. So if we heated up to 60 degrees or higher, we would actually cook the tumor. We call that photoablation. And in fact, we and several other groups have worked on that mode of therapy. And it has some advantages, but it has lots of disadvantages in that it's not really molecularly specific. You know, it's sort of a hammer. In this case, what we did is we just heated up a teeny bit, We didn't want to ablate the tumor. We just heated it up to about 43 degrees Celsius, so about 5 degrees higher than body temperature. And that started the coagulation cascade, so it triggered this network of enzymes that were hanging around in the blood. And at the end of the amplification cascade, the network that results is this protein network called fibrin. And there's a particular enzyme in that cascade whose job it is to cross-link that fibrin. Because when you make a clot, you want the clot to be strong. And so we think of it like a molecular stapler. It goes around cross-linking the fibrin to itself. So what we thought would be great would be to have a drug-loaded nanoparticle now come in and be decorated with a zip code that was sensitive to the enzyme that does the stapling. So the idea would be that you have a drug-loaded nanoparticle come in and it gets basically cross-linked into that newly deposited network. So now you've sort of set off a little reaction with a nanorod, maybe 1 nanorod, and you end up with like 30,000 molecules of drug that have been stapled into your network.
Is this actually what happens? Do you see that large amplification?
Yeah, we actually do see about a 30,000-fold amplification in the deposition of drug.
So that's really impressive. This is so much better than only seeing 1% of a drug going to the target.
Yeah, we're really excited about the efficiency of drug delivery in this system. The sort of extra bonus is it turns out that little extra bit of heat also improves delivery into the tumor, just like when you go out on a warm day and your blood vessels dilate. And we're really also interested in pursuing that in the future because even just the heat without any of the fancy chemical reactions improved our delivery by about 20-fold.
Oh, so just heat alone. So that was a new discovery altogether in this process.
Yeah.
Now, the experiments so far have been done in mice. I'm sure there are people who want to know if they might be able to see this targeted delivery available for humans. Is there a time frame for this?
Yeah, so I think, typically from the first set of therapeutic results in mice to getting to patients is about 5 to 10 years for most things. The path would be that you would license it to a commercial organization and they would work with the FDA to do safety studies in small animals and then larger animals, and then they would start clinical trials. So that's sort of the typical path. In this case, we'd actually like to simplify the system further. We sort of felt like this was just one example of what we hope is a brand new idea of how to have cocktails of nanoparticles that cooperate, and we think it needs to be simplified a little bit before taking it to the patients. In particular, we like to avoid the need to shine the light. We thought it was a really great experimental system and it was really well controlled and it allowed us to see what the effect of a local trigger was. But the downside is that the surgeon actually needs to know where the tumor is to shine the light. And in many cases, in 90% of cancers, the disease has already spread and you don't know where it is. So ideally, you'd like a nanoparticle cocktail that itself can find the tumor and doesn't need to be activated by local light. And we have some ideas about how to do that.
Oh, so there are some superparticles. They sound like superparticles to me that can actually target the tumor and somehow go ahead and initiate a cascade like the clotting system, for example, and bring in the drug.
Yes, so that we definitely have, at least one version of that and another that we're working on. And the trouble there is that you have to worry a lot about safety because now you don't have that extra safety factor of the external trigger. You're injecting something that's going to start clotting wherever it goes, and you have to rely on its specificity for this amplified reaction. And so that's really the trick, is finding something that's very safe and specific in the amplification. And you can start to think about maybe there are other cascades that one might exploit. Clotting was a great first one to start with because of all the reasons I mentioned, but there are actually several others that maybe you wouldn't worry so much about non-specific activation.
So it's a very interesting balance of having this great technology and combining it with the biological system, and in this particular case having a trigger be somewhat innocuous, but then moving on and not having even that trigger, but then balancing it with safety. So there's like let's move ahead in this direction, but wait a minute, because we're taking off that extra step, we now have to be cautious that it isn't going to just affect all cells or cells all over the body, so we have to step back and look in a different direction to be a little more controlled. It's a very interesting balance of safety and simplification along with the technology that you're weighing there.
Yeah, and I think, if you look at technology as a whole, it actually is a theme throughout technology. So, you always start with a really well-controlled system. If you think about robotics as an example, and then ultimately we all fantasize about the autonomous system, the system that knows where it needs to go and knows what it needs to do. And in the minute that you give it all that power, you have to worry about what happens if it goes awry. So, I think you see that throughout technology, and it's definitely something that we're cognizant of.
That's fantastic, and I really appreciate the time you've taken to talk with me about this new work. Good luck with the future efforts. I look forward to talking with you about that soon.
Great, me too. Thanks for having me.
That's it for this week's show, but be sure to come back next time for more of the latest in science and technology. Podcast is a production of Current Science and Technology at the Museum of Science in Boston, part of the Boston community for over 175 years. For more information, visit our website at www.mos.org/CST or email us at podcast@mos.org. Thanks for listening.